At first, there was nothing. From the nothing came something curious. And then we had the plasmodium.
Birthed in agar-agar, sustained by oats, it could do many things, such as conduct electricity, create music, solve mazes, self-repair, and map out entire cities. But there was one thing it couldn't do. It couldn't stand light. The quantum symbol of biology doesn't really get along with the quantum symbol of physics. If light could cause it to behave so, what would happen if the process was reversed? What possibilities are born out of the spooky entanglements between slime and shine? In a world where science meets nature, a daring team of Possum students embark on an incredible journey. Their mission? To unravel the mysteries of nature's own quantum computer, the remarkable Physarum polycephalum slime mold. When it's away from its shiny nemesis, it's happy and growing, its critters thick and thriving. But when light walks into its way, it shrivels up, thins, and moves away. We're imagining an entanglement where measuring one fixes the measurement results on the second, where these forces are inversely proportional. This is an entanglement of biology and physics, of absences and presence, and of classical and quantum. Here's how it was done. 1. We have to grow the slime. Let's call it Mr. Mac Mold. 2. We let Mr. Mac Mold forage. It grows, forms patterns, calculate pathways to avoid lit-up areas, and gets to its favorite oats via the quickest route possible. 3. We place our dear Mr. Mac Mold on a surface of randomly focused spots of light. The on or off states of each light corresponds to 0, 0, and 1, 1 values as derived by running a simple entanglement circuit on the KISS kit. We then map the array of 1024 quantum measurements produced by running the circuit on the equivalent 1024 pixels of a 32 by 32 LED matrix. We test it at different sections against a data set of its primary growth states. This determines the state of LED happiness meter. Another instance of light inextricably linked to the state of the plasmodium. Looking at the face can tell you the condition of the slime, and vice versa. The last step is a visual representation of the mold's changing states. These are growing, not growing, and shrinking. The final form, equal parts installation and visualization, is a literal projection of the single-celled enigma that started it all. But why do all this? 1. Because we're obsessed with the macro effects of microorganisms. 2. Effects that have already brought hope into our lives. Our experiment is akin to uncovering a hidden recipe for processing information efficiently. We hope this recipe can then be used to create future algorithms that outperform the ones we use today. Now where do we go from here? 1. Into the world of education. Our curious yet exciting combination of user-friendly tools like Teachable Machine, LEDs, and Petri dishes set the stage for awareness of a creature so single-celled yet so widespread in its potential impact. 2. Learning about quantum slime algorithms and biocomputing is the first step into actually producing something from it. While there are research opportunities, it remains mostly restricted to academia. 3. We're interested in seeing how the future generations approach the pluralism held within a singular cell capable of complex calculations, carrying them all out simultaneously. It's Quantum Nature, brought to you by Frida, MFATD, Skylar, MFADT, and Shristi, MFAD+T.